Welcome back, friends. Welcome to another video tutorial from Shomu's Biology. We've been talking about the nervous system of human body, and actually we are more interested about how neuron functions. We've talked about neuron structure. We've talked about the resting membrane potential in neuron. We also talked about how action potential is generated in neuron. And this action potential is what we call as a nerve impulse that transfer from one neuron to the next, and it propagates along the axon of a neuron. Now in this video we will be seeing how exactly one neuron is contacting the next neuron, that is the neuron-neuron interaction. And not only we'll see the neuron-neuron interaction, but also we will look at how neurons are interacting with tissues like muscle, call it as a neuromuscular junction, because neuron can have neuron-neuron interaction between two adjacent neurons. It can have interaction with muscle known as neuromuscular junction. It will also have interaction with different glands that will regulate the secretion from those glands. So goal for our learning of this topic is to understand the detailed mechanism of neurotransmitter release, diffusion and binding to the postsynaptic receptors, to learn that the action of neurotransmitters depend on the type of receptor on the postsynaptic cell, and to review the location and the function of these neurotransmitters. You know, this synaptic transmission involves the release of these neurotransmitters from the presynaptic cell to the postsynaptic cell, because here we are looking at the interaction between two neurons, neuron 1 and neuron 2. Neuron 1 secretes certain chemical factors known as neurotransmitters. Once released, it will go and bind to the postsynaptic neuron. It will activate those neurons, and those neurons will be stimulated. They will be stimulated by the activity of this neuron. And by this means, this whole process of neurotransmitter release, it ends when the neurotransmitter dissociates from the receptor and is removed from the internal junction between these two neurons, known as synaptic cleft, the region between two adjacent neurons. Now normally the interaction of neuron works like this. You know, neurons have a cell body, dendrites are attached to it, and then it has the axon. At the end it has the branches, and a next neuron begins with the dendrites. So this junction of neuron-neuron interaction goes with axon to dendron interaction. Okay, so let's look at this structure of this neural junction and synaptic cleft, because all the process of interaction will go on in the synaptic cleft, and we'll also see how this whole process works sequentially. This is the overview of the process that we are looking at. We are looking at the events of the synaptic transmission. Okay. Now the idea of synaptic transmission, if you look at this picture, this is the pre-synaptic cell, this is the post-synaptic cell, and this region in between the two is the synaptic cleft. Okay. And you know, these dots are visualized here inside the vesicles. These are neurotransmitters, and they have different functions to play. There are two types of these neurotransmitters available. One type is known as excitatory neurotransmitter, another type is inhibitory neurotransmitter. Excitatory neurotransmitter allows the next neuron to be excited, and the inhibitory one causes the next neuron to be inhibited of its functionality. So what happens here is the action potential continues to generate from the axon hillock through the axon until it reaches the axon terminal, and this is what we call it as a axon terminal. And this is the region where all those vesicles filled with neurotransmitters fuse with the axon terminal membrane that cause the neurotransmitter release to the synaptic cleft. Once they are released, these neurotransmitters can interact and bind with the neurotransmitter receptor on the surface of the postsynaptic membrane or the cell. That will cause the ultimate effect. So the series of events that we're looking here, it simply start with the action potential generation and it reaches the axon terminal. Second is the calcium channel opens. Third, once calcium channels start to flow, that cause the release of those neurotransmitters by the vesicle fusion to the membrane. The neurotransmitters crosses the synapse, which is between the two axon and dendron. Then neurotransmitters bind to the neuroreceptors that triggers the signal in the postsynaptic neuron. These are the sequential event of the whole process of synaptic transmission. So now let's look at each of the stages in details. We start with the process when it occurs in the presynaptic cell, and the process here is neurotransmitter release, right? So what happens there? An action potential in the axon terminal cause voltage gated calcium channels to open up. As these voltage gated calcium channels open, and why they are open? Because of the action potential. Action potential cause voltage gated calcium channels to open up. Due to this opening of calcium channels, there is a calcium inflow inside the cell, presynaptic cell. That will cause all those vesicles filled with neurotransmitter to be fused to the membrane. Now, each vesicle released a fixed amount of neurotransmitters into the synaptic cleft. Now, this neurotransmitters diffuses through this synaptic cleft and it will reach the receptor, bind to the receptor on the surface of postsynaptic cell. Now chemically gated ion channels, due to the binding of this receptor, opens up, okay, because this is not about the voltage. As these channels are chemically gated, they will remain open as long as the neurotransmitter is bound to the receptor and are not sensitive to any changes in the membrane potential. So irrespective of the membrane potential, if the chemical is attached to the receptor, it will remain open. Now the synaptic current or ion movement through the chemically gated channels may depolarize or hyperpolarize the neuron. Now due to the change, whether it can depolarize or hyperpolarize the postsynaptic cell or postsynaptic neuron, there are different events and effects that we can see. The second step occurs to the postsynaptic cell. That's known as the receptor binding. When the neurotransmitter is released, now those neurotransmitters will bind to the receptor on the surface of the postsynaptic neurons. Okay? Chemically gated ion channels will open up because these ion channels are also closed normally without this chemical neurotransmitters. But whenever the adjacent neurotransmitters are attached to this receptor, they remains open. Now opening of this receptor will cause other types of ions to flow in or out. That can be sodium, potassium or any else. For example, here you see, due to the attachment of this purple colored neurotransmitter to the receptor, it caused the potassium to flow out and sodium to flow in, right? Now these chemically gated ion channels will remain open as long as the neurotransmitter is remain attached to the receptor. And these are not sensitive to any changes in the membrane potential. Even though the membrane potential is changed, still this will function based on the attachment of neurotransmitter only. Now the synaptic current or ion movement through the chemically gated channels may depolarize or repolarize or hyperpolarize these neurons. This example that we see in this picture explains this whole process of how it can depolarize or repolarize, because as the channel is open, it can transfer the positively charged ions across. That can change the membrane polarity, right, and the polarization of the membrane. Now the third part of the synaptic transmission is the termination of synaptic transmission, because all this process should have to have an end. Now synaptic transmission ends when the neurotransmitter dissociates from the receptor. This is when the neurotransmitter is attached to the receptor, they make these channels always open. But whenever the neurotransmitters are released, these channels are closed. Now most often the neurotransmitter is pumped back into the presynaptic terminal and into the nearby glial cells. Here we illustrate the neurotransmitter glutamate being pumped back into the from postsynaptic to the presynaptic neuron again. Okay. In some cases you see the neurotransmitter is broken down by enzymes and the products are pumped away. So either they can be degraded or they can be pushed back from where they are released. Okay. Now the neurotransmitter acetylcholine is an example of this process. When the process is done, the neurotransmitter is broken down. When breakdown products are transported into the presynaptic terminal, they are used to resynthesize the neurotransmitter again. So simply break them down after the work, then again we take the breakdown copies and shift it to the presynaptic neuron so that they can engulf and take all those breakdown content of the neurotransmitter and can resynthesize those neurotransmitters. The neurotransmitter which has been returned to the terminal is packaged into the vesicles for storage and subsequent release in the future. Okay. And by the mechanism which this neurotransmitter is returned to the terminal is specific for each neurotransmitter and can be selectively affected by different drugs. So we can use this as a drug targeting approach for a range of drug targeting and drug development. Okay. Now final thing is the type of response from the postsynaptic cell due to the attachment of the neurotransmitter. Now we have seen that the mechanism of synaptic transmission, the sequential stages, we are talking about that. If you look at the consequent synaptic activity after the release of neurotransmitters and binding of neurotransmitters, that is the ultimate goal that neurons need to achieve. Now the action of these postsynaptic cells depends on which neurotransmitters is involved with the signal. These are the neurotransmitters that we are looking. There are multiple receptors for each neurotransmitters present on the surface of this postsynaptic neuron. Each of these receptors activates a different set of ion channels. Some of the receptors may activate sodium channel, some of them may activate potassium channel. Now depending upon which type of ion channel it opens, the response varies. There are two groups of receptors that mainly find out. We can find it out. One is a cholinergic receptor and another one is adrenergic receptor. Let us look at different type of receptors. We will see two types of receptors. We will see the excitatory and inhibitory receptors. One of the excitatory receptor is this acetylcholines, and we will look at the acetylcholine as a neurotransmitter and its associated receptors. There are multiple types of receptors for each of these neurotransmitter, and they are associated with the activity or activation of different ion channels in the cell. They can activate sometimes sodium channel, or in some case some other neurotransmitter can activate the potassium channel. Now there are two groups of these receptors associated to the acetylcholine. One of the receptor is a cholinergic receptor, another one is chemical nicotine. So these cholinergic receptors bind to the acetylcholine. Cholinergic receptors bind to the acetylcholine. On the other hand, the other group that is present there, one group also bind to the chemical nicotine and another group is there that can bind to the chemical muscarine or muscarine. So these are the three types. This is where we see that the acetylcholine receptors bind with the nicotine. This is where it binds with muscarine, and this is one is also example of a muscarine. This cholinergic nicotinic receptor, or it's also known as NACH, cholinergic nicotinic receptor, is a very well known receptor found in the neuromuscular junction. For example, where the neuron is interacting with a muscle, and the release of neurotransmitter regulates the contraction and relaxation of the muscle. Okay. At this receptor, acetylcholine acts as directly to open the ion channels, producing a fast excitatory postsynaptic potential. Very fast activity is going on there. Acetylcholine is excitatory at nicotinic receptors, and it causes skeletal muscle to contract. So this is the reason that you can, for example, you're walking, suddenly your foot, your toe is somewhat attached to, or let's say in touch with a very hot plate or something. Suddenly your hands or toe, whatever is touching to the hot plate, is opening up, it's closing down. So due to the muscle contraction, these things happen. While the other type of the cholinergic muscarinic receptors, so we call it cholinergic muscarinic receptor, all known as MACH, cholinergic muscarinase or muscarinic receptor. This is found in central nervous system most of the time, and most of the effector organs of the parasympathetic branch of the nervous system, because here acetylcholine acts indirectly in those systems. Because you know, here is the difference between the two. We have a situation where acetylcholine can act directly, which is very fast activity, and another situation where acetylcholine can act indirectly, which is very slow activity. But the slow activity can be of two different types, excitatory or inhibitory, and those things are required in the central nervous system, in sometimes to influence or sometimes to inhibit the response. Okay. Now this acetylcholine is excitatory at this muscarinic receptors and cause neurons to fire action potential and the smooth muscle cells to contract. It will cause the smooth muscle cell to contract. Okay, but this activity is little slow. But another type of MACH receptor is also found in the central nervous system and as well as in the heart. This acetylcholine acts, here in this case the acetylcholine acts indirectly and producing a slow inhibition of the postsynaptic cell. Here it causes the inhibition. In the heart, this effect decreases the heart rate, and acetylcholine is inhibitory at this muscarinic receptor and causing neurons to hyperpolarize and the heart to slow down. The action of this acetylcholine may be excitatory or inhibitory, but the effect depends on which receptor is present on the postsynaptic cell. If it is a type of nicotinic receptor, it's always excitatory and it is very fast. But if this acetylcholine is binding to a muscarinic receptor, there are two chances. One chance that the muscarinic receptor binding will excite, that happens in most of the cases of smooth muscle contraction, or in some case binding of acetylcholine to this muscarinic receptor can inhibit and can cause the heart rate to slow down due to the hyperpolarization. Okay. Now let's look at norepinephrine and its receptors. This is another very important neurotransmitter, norepinephrine. There are two families of receptors for neurotransmitter norepinephrine. One is alpha receptor, another one is the beta type of receptors. Each family members is different and they are identified by its letter and number. Alpha 1, for the beta there is beta 1 and 2. Okay, these are called as adrenergic receptor. Remember we talked about nicotinic, muscarinic, and this is adrenergic receptor. So all those nicotinic and muscarinic are the part of cholinergic receptor, but this is a completely different type known as adrenergic receptor. Both alpha and beta adrenergic receptors are found in the central nervous system, and more importantly on the effector organs of the sympathetic nervous system. In this case, this norepinephrine acts indirectly at alpha 1. If you look at the case of alpha 1, it works indirectly to the alpha 1 receptor, and it causes slow excitation, because we know if the process is indirect, always it's slow, right? This causes smooth muscles to contract, and alpha 1 receptors are located mostly on the blood vessels, as you see in this picture, which supply the skin, mucous and abdominal viscera. This norepinephrine is excitatory at alpha 1 receptors, so it excites them, but it's a slow excitation. Now, as I also told you, there are also other types, beta 1 and beta 2. So if you look at beta 1 type now, beta 1 is another receptor, and when norepinephrine interacts indirectly to beta 1 receptor, that is mostly in the heart, to produce a slow excitation, and in this case it will allow the heart rate to increase, because this is also a slow excitation. And norepinephrine is also excitatory at this phase, but slow. The third one is when the norepinephrine binds indirectly to beta 2 receptors, and that will also produce a slow inhibition now. So alpha 1 and beta 1, both the receptors when added to or attached to norepinephrine cause a slow excitation, but whenever norepinephrine is attached to the beta 2, it causes slow inhibition. This type of interaction of norepinephrine to beta 2 receptor produces slow inhibition that causes the smooth muscle to dilate. These beta 2 receptors are mostly located on the respiratory airways, blood vessels and skeletal muscle and heart, and most of the other effector organs of the sympathetic system. Norepinephrine is inhibitory at these beta 2 receptors, and as a result the action of the norepinephrine may be excitatory or inhibitory based on which type of receptor they are binding. So one thing we can say for all this type of neurotransmitter is that there is a bunch of neurotransmitter and their associated receptors are found, and those receptors are found in different regions and different organs and tissues of our body. Now, how the cell will react is denoted by the receptor where the neurotransmitter is binding. A same neurotransmitter can cause inhibitory or excitatory effect depending upon the variant of receptor they are binding, and that is true for both acetylcholine as a neurotransmitter and norepinephrine as a neurotransmitter. Now if we'll talk about the location and function of these neurotransmitters, we have learned about acetylcholine and norepinephrine, and we found out that the central nervous system, and we found them in central nervous system and also in effector organs of the nervous system. Now here we will see all the other neurotransmitters of the peripheral nervous system, because that's also very important, right? So then we'll look into the central nervous system to learn the functions of these and other neurotransmitters what are working there. So you see here the effector organs of the nervous system like skeletal muscle, smooth muscle, cardiac muscle and glands. All these things are regulated by those acetylcholine and norepinephrine. But if you look at the peripheral nervous system and how these neurotransmitters are working in the peripheral nervous system, you will see that the motor neurons of the somatic nervous system release acetylcholine, okay, and they are cholinergic. So skeletal muscles bear NACH, which is nothing but the cholinergic nicotinic receptors. So as skeletal muscles contain these nicotinic receptors, the action of this acetylcholine on skeletal muscle is direct, and fast. So if we're talking about the somatic neurons, they will work really fast and they will work excitatory by the release of acetylcholine because they only have cholinergic nicotinic receptors. The first of the two neurons in the sympathetic chain, if we look, preganglionic neuron, are cholinergic. These first two neurons in the parasympathetic chain, the preganglionic neuron is also cholinergic. While the second neuron or postganglionic neuron in both the sympathetic and parasympathetic chain, all of them have these NACH receptors. Thus, the action of acetylcholine on postganglionic neuron is direct, fast and excitatory. So you look at here, for postganglionic neurons it's always excitatory, it's always fast for both sympathetic and parasympathetic neurons. But for somatic it's different. In somatic we also talked it carries NACH, but postganglionic neurons carry NACH, so they will also have a fast response. We know that these somatic cells or somatic neurons connected to the effector organs like skeletal muscle, while this sympathetic and parasympathetic part of the autonomous nervous system, if you see, they are linked with smooth muscle, cardiac muscle and gland. Let's move to the next one. If we look at the sympathetic postganglionic neurons, they release norepinephrine. If we go back here, sympathetic postganglionic neuron, they release norepinephrine and they are adrenergic. Parasympathetic postganglionic neuron release acetylcholine, so they are cholinergic. Both this acetylcholine and norepinephrine act indirectly on the effector organs of the autonomous nervous system. Their action is slow, sometimes excitatory, sometimes inhibitory, but it completely depends on which receptor is found on the surface of the effector organs like skeletal muscle, smooth muscle, cardiac muscle or gland. So they can either inhibit or excite depending upon which receptor is provided on the surface, and especially which receptor is provided in the postsynaptic neurons. If we talk about the excitatory and inhibitory neurotransmitter in CNS, in this case in central nervous system, glutamate is the most common and most potent excitatory neurotransmitter. Glutamate, simply write it as GLU. Glutamate acts directly on the ion channels that permits the passage of both sodium and potassium movement that produce a fast excitatory postsynaptic potential, and the major inhibitory, so this is the major excitatory, so the major inhibitory neurotransmitter in the central nervous system is GABA, gamma aminobutyric acid, and glycine. GABA and glycine binding to the receptor which directly opens chloride channel, which completely reverses the mechanism of glutamate action, because chloride channel will transfer negatively charged ions across the membrane. It causes fast inhibitory postsynaptic potential. In central nervous system we need these fast actions, because you know in controlling all our smooth muscles it's always not fast. We don't require those things to work faster always. Simple modification, sometimes you also need slow modification, but in CNS we need fast actions. That's why we have glutamate as a positively influencing neurotransmitter and GABA and glycine as a negatively influencing or inhibitory neurotransmitter. If we look at the fast versus the slow synaptic action, there is a difference between the direct and indirect activation of the neurotransmitters. You know, the rapid signaling of the directly acting neurotransmitter is important for the secondary motor coordination and communication and many other higher functionalities. So they help in the sensory motor coordination, communication, higher functions. For example, rapid synaptic signaling is essential for coordinating sensory output and input together. Sensory input along with the motor output, especially during the athletic performances, that helps you to do this job. Rapid synaptic signaling is essential for speech and other form of communication, and also important for playing music and requires rapid synaptic signaling. On the other hand, all the neurotransmitters that affect the organs of the peripheral autonomous nervous system act indirectly. Those are the indirect activations. Norepinephrine, acetylcholine and serotonin, another central nervous system neurotransmitter, all produce state changes in the central nervous system. One important example is the change from the sleep state to the wake state. That is one thing, and that needs to be done slowly. That is known as a wakefulness or arousal. It needs to happen slowly, not that rapidly or fast. On the other hand, theories of learning and memory often invoke the action of indirect neurotransmitter to explain the changes in synaptic activity. These indirect neurotransmitters can modulate neurons and ultimately changing their channel function and producing a new and different types of output, and that is important for learning and for a long-term memory and the activities associated with memorizing something. So it's not a rapid task. It will build up slowly but steadily. So that in a sense are the things that we want to talk about. So let's look at the summary of what we've learned. We've learned so far that the neurotransmitter is released from vesicles in the presynaptic cell and binds to the receptor of the postsynaptic cell. Neurotransmission ends when the neurotransmitter dissociates from its receptor. The effect of neurotransmitters on the postsynaptic cell depends on the receptor, not the neurotransmitter molecule. If we vary the receptor, the response will also vary. Sometimes the response is inhibitory, sometimes it is excitatory. Acetylcholine and norepinephrine are the most important neurotransmitters in the peripheral nervous system, while glutamate, GABA and glycine are the most important neurotransmitters in the central nervous system. Glutamate work as a excitatory neurotransmitter in CNS, while GABA and glycine work as an inhibitory neurotransmitter in the CNS. And fast and slow synaptic activities serve different functions. Fast activity is required for the athletic performances. It is also required for the communication skills and other higher functions and sensory motor coordination, while the slow activity required for the learning and memory. That in a sense is the gist about this topic. I hope you understand the video. If you like this video, please hit the like button, subscribe to my channel to get more and more videos like that, and share this video with your friends. Thank you.